everyone, and welcome to today's Drug Target Review webinar titled Pain Research, WRPRFa as a Novel Tool for Acid Sensing Ion Channel 3. My name is Sam Whitewick, Editorial Assistant at Drug Target Review, and I'll be moderating today's webinar. On behalf of Drug Target Review and Eurofins DiscoverX, I'd like to thank you all for coming today, and I'm happy to be joined by Dr Nien-Du Yang and Dr James Costantin as they present new research on WRPRFa, a potent and selective peptide modulator for ASIC3. Following the presentation, we'll move on to the Q&A session, so please submit your questions at any time during the discussion via the questions panel on the right-hand side of your screen. And with that, I'll hand over to Drs. Yang and Costantin to begin the webinar. Over to you guys.
Today, we are pleased to welcome Nien-Du Yang of Latigo Biotherapeutics to present data on a peptide tool that they developed that affects the properties of the ASIC channel. He'll be giving the background of the ASIC channels, discovery, characterization, and application of a peptide that they developed, and then I could be going over our ion channel products from Eurofins DiscoverX, including our capabilities and the products that we offer. And with that, I will hand the presentation over to Nien-Du. So, thank you very much.
Hello, thank you, Jim, for the introduction. My name is Nien-Du Yang from Latigo Biotherapeutics. Today, I'll present on a peptide that we developed in-house and how we use it as a tool to understand ASIC biology for pain research. And first, I'll some backgrounds on ASICs and how we got to this peptide. So ASICs are acid sensing ion channels that are activated by extracellular protons. As we see in the structure here, ASICs are trimeric channels where three subunits colored differently here come together and form either as homomers or heteromers. They have a very large extracellular domain that contains an acidic pocket critical for proton sensing. And upon a decrease in extracellular pH, this domain undergoes conformational changes that ultimately lead to the opening of the ion conducting pore in the transmembrane domain. This representative trace here shows how ASICs respond to a low pH stimulus. So from a resting or closed state, the channel activates and opens rapidly. Once the channels open, it allows mainly sodium ions flowing into the cell, so an inward current here, and that depolarizes the membrane. And it is followed by a process called desensitization, where the channel closes in the presence of the stimulus and remains closed until they are recovered at physiological or more alkaline pH to be activated again. ASICs are involved in various physiological and pathophysiological processes, but the most important thing for us as a pain company, ASICs are found in the, pain sensing neuron called nociceptors in the dorsal root ganglion, DRG, making them an attractive targets for analgesia. And here's some evidence that ASICs are expressed in human DRG neuron. On the left here is a set of traces showing a human DRG neuron stimulated with increasingly acidic stimuli from pH 7 down to pH 6.3. And we can see that these neurons can sense relatively mild acidification by directly evoking action potential, which we can think of as the currency of pain, or by depolarizing the cell membrane to increase excitability to other stimuli. These traces on the right here shows that application of an ASIC inhibitor dimenosine that decreases and block the acid evoke action potentials. So these indicates that these responses in human DRG are driven by ASICs. And there are 4 genes in the ASIC channel family, ASIC1 to ASIC4, that along with alternative splicing give rise to a total of 6 different ASIC isoform, and several of them are actually found in human DRG neuron. Here on the top here, I'm showing RNA-scope images showing ASIC1, ASIC2, and ASIC3, as well as ACN10A, which encodes NaV1.8 in human DRG. Bottom left here is an RNA sequencing data from human DRG of several donors, showing that ASIC1 and ASIC3 are actually the two predominant isoforms, with a little less of the ASIC2. And if we take a closer look at the RNA scope analysis shown in the pie chart here, we see that the majority of cells are either expressing ASIC1 only or co-expressing ASIC1 and 3. What is not shown in the pie chart, but it is obvious from the RNA scope images, you can see that the ASIC1 and ASIC3 expression overlaps pretty well with NAV 1.8, which is a biomarker for nociceptors. For the cell population that expresses both ASIC1 and 3, an interesting question arises here is whether they're actually forming separate homomeric channels or heteromeric channels. And do these channels play different roles in pen sensing? And actually, we found that these different ASIC isoforms have distinct properties in their gating. So here's a set of traces for ASIC1A, ASIC2A, and ASIC3 currents evoked by different pH stimuli, increasingly acidic, from the left to the right. We can see that ASIC1A and ASIC3 have a very similar sensitivity to proton activation in that they activate at a similar range of pH. However, you can see from the traces that ASIC3 desensitize or closes much faster than ASIC1A across the board. ASIC2, on the other hand, requires a very acidic pH for activation in that they don't really open until you get down to pH 5. So each of these isoforms has these unique properties that can come into play when making a complete ASIC trimer. For example, you can imagine the properties of a heteromer channel can be tuned by its subunit composition. But the real question here is how do we tell these different ASICs apart in tissue? So what we really need are pharmacological tools that can help us discriminate between these different ASICs. Here we focus on ASIC1A and ASIC3 since those are the two main isoforms in human DRG. This graph here shows the ASIC1A versus ASIC3 potency for a number of inhibitors from either patents or literature. I want to specifically mention the team at Matrion led by Gary Clark and Andy James for their great work generating this very nice data set here. This red dashed line here represents an equal activity against ASIC1A and 3. And we see several points here on this line indicating there are non-selective inhibitors. We see below this line some inhibitors that are a bit more ASIC1A selective, but we don't really see on the opposite side that are selective for ASIC3. From what we know and not shown here, that there is a toxin out there, Apex2, called Apex2, that selectively inhibits ASIC3, but it also hits NAV 1.8, which you can imagine would be a problem for our functional study looking at ASIC in human DRG. So what we really need here are better ASIC3 selected tools. We then turn our attention to activators and found that there's actually an ASIC3 selective activator out there. There's a peptide from corn snail venom called WRPRFa amide. So it's a tetrapeptide with an amidated C terminus. So I'll call it RPRF for simplicity for the rest of the talk as it's much easier to say. It's been reported that RPRF selectively slows the desensitization of ASIC3 to prolong channel opening and therefore gives the activating effect. However, the effect of RPRF are quite modest and transient. But we think it's a good start here, but we wanted to see if we could build on it and do a better job. Next, I'll walk you through how we discover this new peptide. So the path we took was to do a broad structure activity relationship or SAR screen First, to understand the relative contributions of each amino acid of RPRF to its activity. So in order to do this in a higher throughput way, we developed an ASIC3 assay on the Sofian Q-patch automated patch cleanse system using Eurofins DiscoverX, a human ASIC3 stable cell line. So as you see here, the cell line performed quite well on our Q-patch assay, as you see on the left here. Robust ASIC3 currents evoked by our standard pH 6.3 stimulus. And the currents are also pretty stable across multiple liquid periods here, which is always nice to see on an automated patch cleanse system. We then tested our tool compound deepening, shown on the right, and the potency value came out matching what's been reported in the literature. So at this point, we have a robust assay ready to go. Next, we design a Q-patch protocol shown here on the top screen to screen for peptide activity on ASIC3. So what we do is we do 2 vehicle pH 6.3 first, and then we incubate the cells with 3 or 30 micromolar of peptide at pH 8, followed by application of pH 6.3 again. And the trace here in the bottom shows RPRF as an example. Since ASIC3 fully desensitized in 3 seconds after activation, and so there's basically no currents at 3 seconds in the vehicle. So for the peptide treatment, we use the current remaining at 3 seconds and normalize that to the peak current to approximate the fraction of the channels bound to the peptide. And this ratio I 3 seconds over I peak is used as the measure of peptide activity on ASIC3 in our screen. We generated a panel of custom peptides with variation and changes to the RPRF and put them all through this Q-patch screening assay. Okay, although we did an entire SAR study around RPRF, for the interest of time, I only shown here a small panel of our screen. So basically what we found was by adding a tryptophan to the N-terminal end of RPRF to make WRPRF produces a striking increase of ASIC3 activity as shown here in the traces and also in the ratio graphs here. I wanted to mention that adding other aromatic ring-containing amino acid to the N-terminal also generates a large effect, but the effect was most pronounced with WRPRF. So that's the peptide we continue our work with. And how does this new peptide compare to its parent RPRF? So here we did a concentration response from 0.3 micromolar all the way to 100 micromolar for both peptides. And the potency curves are shown here on the right. So what we found is that WRPRF is a much more potent peptide and also had a larger maximal effect compared to RPRF. It is also obvious from the traces that WRPRF dramatically slows ASIC3 desensitization and produces these long-lasting channel openings. So now we have this improved and more effective peptide WRPRF. We wanted to characterize it a little bit more. So next, I'll be talking about some of the properties of WRPRF and demonstrating how those properties can make this peptide a useful tool in certain applications of ASIC. The first property is pH sensitivity to activation. Here we did a pH response for ASIC3 in the absence and presence of WRPRF. And the pH dependence of activation curves are shown here on the right. So what we found is with WRPRF treatment, the halfway point of activation, which we call the pH 50, was shifted in the alkaline direction from vehicle to around neutral pH. meaning that the channel activation now is more sensitive to proton. This is also obvious from the traces below with WRPRF treatment that you see ASIC3 already activates at pH 7, in addition to the prolonged channel opening effect compared to the vehicle. And how does this property translate in neurons? So here we tested WRPRF on both human and rat dissociated DRG neurons. So we stimulate the neurons with increasingly acidic stimuli from pH 7 to pH 6.3, and we are looking at in current clamp in the absence and presence of the peptide. And we found that WRPRF, as you see, produce a larger and sustained membrane depolarization than the vehicle at less acidic pH, and in some cases even evoked more action potential shown as the numbers below. These are consistent with WRPRF increasing DRG neuron activity by making them more sensitive to ASIC stimuli. So one other interesting observation here is that you see the degree of the depolarization as well as the number of action potential evokes varies dramatically between neurons. And I'll touch on why that is later. The second property is selectivity. So we wanted to know whether an increase in the activity on ASIC3 actually reduces its selectivity. So, but what we found was that WRPR still maintains a very high selectivity for ASIC3 over ASIC1A and ASIC2A. And in our hands, it's even a little bit more selective than RPRF, which shows some effect on ASIC1A. With this kind of high selectivity profile, it allows us to conduct a census of ASICs in dissociated neurons in vitro. And we did that for both rat and human DRG neurons. So the way we did these experiments is we first apply a pH 6.3 stimulus to each neuron. If it evokes a current, it's marked as ASIC positive. We then applied the peptide, and the cells responded to the peptide WRPF are marked ASIC3 positive. And the major difference we saw between rat and human is first that only 1/3 of the neurons in the rat have ASIC current, while every single human DRG neuron we tested all have ASIC current. Another thing we noticed is that every human neuron also responds to the peptide and therefore are ASIC3 positive. While that is not the case in rat neurons where we see a portion of non-responder to the peptide. However, the ASIC3 positive here does not necessarily mean the neurons have purely ASIC3. It could only mean that the ASIC3 subunits are present there. And that brings up the next property I want to talk about is WRPRF activity on heteromers. And since ASIC1 and ASIC3 are the two major isoforms in human DRG, we created 131 and 313 concatemer and tested our peptide on these heteromers. So the results showed that WRPRF actively titrate with the number of ASIC3 subunits. So with ASIC3 homomer having the largest effect with 3 intact subunit, 313 in the middle with 2 ASIC3 subunit, and 131 having the smallest effect with just 1 ASIC3 subunit. And we see the potency graph here of that the potency follows the same trends with 131 being the least potent of the 3 channels. With that in mind, and looking back at the DRG neuron data, here each data point represents a response of each individual neuron. We see that for the human neurons actually have a wide range of responses to WRPRF. There are cells with a very strong response like this, suggesting there is a high fraction of ASIC3. And there are also cells with a much smaller response like this, which is perhaps more of an ASIC1 biased neuron. In the red neurons, although it's a much smaller data set here, the response seems to be more polarized. And we also analyzed the desensitization kinetics of ASIC currents in both human and rat neurons. And we notice in human neurons, the kinetics are pretty consistent. Where in the rat DRG neuron, we see kind of a two components and a fast and a slow. So these observations together suggest that there's maybe a difference in the ASIC composition and distribution between human and rat, that the ratio of ASIC 1 to 3 is in rat neuron is more extreme than in human, meaning that you may get more cells that have ASIC 1A homomer than the heteromers population. The last property I want to talk about is probably the main difference between WRPRF and RPRF, which is the effect on desensitization. As you see here, although RPRF slows ASIC3 desensitization, the channel still fully closes after 20 seconds. On the other hand, WRPRF effectively removes this desensitization process, and the channel remains stably open for the duration of the stimulus. It can go as long as before the cell dies or we lose seal. It's been shown that dimenosine is an open channel blocker that binds to the open pore of ASICs. So here's a trace of ASIC1A. So we activate ASIC1 channel with acid, and then halfway through the activation, we apply dimenosine, and it rapidly blocks the current much faster than the channel desensitization. However, you can already tell that it's a little bit quite challenging to measure the kinetics of the block due to the desensitization process taking place. And you can imagine it's even harder with ASIC3 that I showed before, desensitized so much faster than ASIC1A. However, this is now possible in ASIC3 with the help of WRPRF, which essentially creates a constitutively open channel. And during this open phase, we can apply dimenosine that blocks the channel, and then we can also wash the compound off, allowing us to measure the kinetics of on and off rates. This is useful for comparing different pore blockers. For example, this blocker from Merck here, you see, actually have a much slower off rate compared to the other pore blockers we tested. This property of WRPRF could also provide insights into how ASIC inhibitors, even with a different mechanism, work, and how all that might be relevant for blocking acid-evoked action potential and pain. So to summarize, through a peptide screen, we identified WRPRF as a more potent and effective activator of ASIC3. We showed that WRPRF can enhance the activity of DRG neurons under mild acidification, presenting a potential in vivo tool for future ASIC studies. The high selectivity and heteromer activity of WRPRF allows us to probe native ASIC channel composition and distribution. And lastly, WRPRF modulation of gating can provide additional insights into how ASIC channel work and their pharmacology. With that, I want to conclude my talk to thank the Latigo members that are involved in this work, especially John Gilchrist that oversees our internal ASIC work, and Jerry and Vicky who did a lot of the biophysical characterization in the neuron work. I would also like to thank the external collaborator GenScript for making all our peptides, and Abios for providing human DRG neurons and Metrion. Thank you. And now hand it over to Jim for his presentation.
Thank you, Nien-Du, for that wonderful talk. I'm going to present a little bit about Eurofins DiscoverX and our ion channel products. DiscoverX was founded in 2000 and became the products business of Eurofins Discovery in 2017. We provide over 1,000 cell-based functional assays for popular target classes, including GPCRs, kinases, and the others listed here, as well as ion channel products that I manage. Our ion channel products are suitable for target discovery, hit screening, lead optimization, and safety studies. We offer stable cell lines for the ion channels. We also have been converting our stable cell lines into ready-to-assay frozen cells, so RTA cells. We also offer cell-based trafficking assays for ion channels and other protein targets. And we have custom development capabilities in case you would like an ion channel developed that is not in our portfolio. The precision cell lines have an interesting history, so they go back to the early 2000s, where I actually worked on them in the early days with Cytomics. Cytomics began the collection of the cell lines. The cell lines then went from Cytomics to Serologicals to Millipore to Merck Millipore, and as I mentioned, well, Eurofins acquired Merck Millipore, and then they acquired DiscoverX in 2017. And then that's when we became the products company for Eurofins Discovery. The actual stable cell lines that we offer are listed here. So we have quite a few cell lines, a lot of potassium channels, sodium channels, and other channels, including the ASIC channel. And right now we have a total of 60 stable cell lines that we offer. And we validate them on the SynchroPatch 384 well system. Here I'm showing some data from the SynchroPatch system on the validation of the ASIC cell line. And so this is basically the addition of pH 6.0 external solution to activate the ASIC channel. And then we use a non-specific compound amiloride, I should say non-specific, not as specific as the peptides that was just presented, to show block by amiloride of the ASIC3 channel. We also offer these RTA cell lines, as I mentioned, or RTA cells that we converted from our stable cell lines. So far, we've developed 9 of them, and that's the cardiac safety panel, including HERG, CAV 1.2, and AV 1.5. We offer some pain targets, including NAV 1.7 and 1.8. We also are offering nicotinics, and we're launching these others soon. We'll be launching 9 and most likely 10 by the end of the year. We have the data for the first 3 here, and we just have to validate the last TRP channel here to launch by the end of the year, the new RTA products. I'm showing data from the products that have been released already, and they're typical of what you would expect for the particular cell line. And the nice thing about the RTA cells is that they do not require any cell culture. You just crack the vial open and put them on your recording rig or automated electrophysiology platform, and you're ready to go. And with that, I'd like to wrap up this webinar, and again, thank Nien-Du Yang for his wonderful presentation, and thanks everyone for joining us today. Thank you.
Thank you both for those fantastic presentations. As I mentioned earlier, we'll move on to the final part of today's session, which is the Q&A session. A quick reminder that you can still submit your questions using the questions panel in the menu on the right-hand side. But with that, let's get started. So the first question is, where is the binding site for WRPRF?
Oh, okay. Thank you for that question. So we do have data looking at the putative binding site of WRPRF, and from our peptide mutagenesis screen, SAR, we learned that the 2 arginines and the positive charge in the peptide are pretty important. So if we neutralize the charge, we see a big decrease in activity. So we then examine a few counter charges as potential interacting partners, and we did that by using mutagenesis and double mutant cycle, and we identify a glutamate pair in the lower PON domain of ASIC that's very near the beta 11 and 12 linker that is critical for the desensitization of ASIC channel that forms part of the peptide interaction site. And I also want to mention that these glutamates that we identify have also been shown to be important for interaction of other RF emitters like FRRF and RPRF with ASIC1 and ASIC3 respectively in their docking and mutagenesis studies. So kind of the results confirm the functional coupling between the glutamates in the lower palm domain of ASIC and with the RFM at core arginine. But we recognize that additional residues may be involved in WRPRF binding.
Great, thank you for that. And the next question I have here is, RPRF has been used to induce pain in rats. Would WRPRF be expected to do the same?
Okay, that's a very good question. So we haven't tested this in vivo yet, but other studies have, inject this peptide into like the mice leg muscle and show that it strongly enhance the acid-induced muscle pain. So from an in vitro cellular assay, I would expect it probably will do the same by increasing ASIC activity and also increase neuronal excitability and firing action potential. But as you can see in one of the slides I have, that if there's too much of the ASIC activation, it may enters something like a depolarization block. So you actually get a fewer action potential with a very strong effect. But other thing that uh, I want to mention is the value of our SAR study is that it is known that some cationic peptides, like those with a lot of arginines, and can cause mast cell degranulation that may be misinterpreted as effect of the peptide. So for an RSAR study, which is not shown fully here, it could provide some like the inactive analog that will have similar identical charges, but without any as ASIC3 activities as a as a control.
Thank you. The next question we have is just a technical question on neuropharmacology about the figure with DLZE-EC50. What is the Hill coefficient there? I don't ask about precise numbers, just approximately. And does it change in homomeric versus heteromeric ASICs? Thanks.
Okay, I think you mean the audience probably mean the dimerizing IC50. So we have tested that on ASIC1, ASIC3, and the heteromer cell line 2. It is the Hill slope is around 1, so there's not much difference. Also, the ASIC1 and ASIC3 share the same conserved pore binding site with for dimerizing.
Great, thank you. And the next question is, are the ready-to-assay cells division arrested, so not in continuous culture format?
Yeah, our ready-to-assay cells are division arrested using a Eurofins DiscoverX proprietary method.
Great, thank you very much. Next question we have here is, given the difference in ASIC functional expression between human and rats, is the rat data translatable to humans?
Okay, so there's quite for our DRG census, there's quite a big difference between rat and human. So the first kind of difference is in human, they all have ASIC, where in rat, probably only like 30% have. So there's a lot of different possibility why that is. So it could be, the rat has less ASIC, could be an artifact of the dissociated process itself, or it could also reflect an intrinsic distribution difference between human and rat. That would suggest the two species are using ASIC a little differently. So rat may not be the best model for a human in terms of ASIC. So I would say, and then the other aspect is that although they'll have ASIC3 in there, but we saw a difference in the composition of 1 and 3, or the distribution of 1 and 3, where in red there be maybe more polarized or the homomer than the heteromer. And we do see in rat and human different kinetics of the ASIC current. We have not fully linked that to the effect uh, of the WRPRFa to look at whether those difference in kinetics linked to the amount of ASIC3 yet. But I think the effect that the fact that these ASIC1 and ASIC3 biophysically have different desensitized kinetics will seems to be assuming that there's some reason that these neurons have two different isoform present. And given that the human and rat show a difference in kind of the relative abundance of 1 and 3, maybe suggesting that there's probably a functional reason to have 1 versus 3. And there should be some difference between human and rat in the roles of ASIC. So I would say some aspect of the rodent stuff data may be translatable, but given the neuron difference that we see, it may be there's a lot of caveats to that.
Thank you, Dr. Yang. The next question I have here is, there was a striking difference in effect of WRPRF peptide on AP firing in rats versus humans DRG sensory neurons, with massive firing at pH7 in rats not seen in HDRG. Can you explain why? For example, is there a species difference in peptide affinity or pH dependence of ASIC isoforms across species?
Sorry, I got a little caught off here. Can you repeat that question?
Yes, no problem. There was a striking difference in effect of WRPRF peptide on AP firing in rats versus human DRG sensory neurons, with massive firing at pH7 in rats, not seen in HDRG. Can you explain why that is? For example, is there a species difference in peptide affinity or pH dependence on ASIC isoforms across the species?
Okay, we have not tested the pH 50 of rat isoform versus human, but I think from the literature they're pretty close and they may not show a difference there. But I think from a neural perspective, we only tested quite a few, and as you see, the data kind of varies a lot, so it's a little bit hard to tell at this point with the the current clamp data that would suggest any difference with from the peptide experiment.
Thank you. Next question is, the pH of the bath solution was 8. Did they test if the effect of WRFA is dependent on the pH prior to activation?
Yes, so so I think you're getting to a question of whether the WRPR binding is sort of state dependent. So we have some data to show that the WRPR preferentially interact with the closed state, which is pH 8, around pH 8 that the channel fully closed. And we have data to show that it preferred the closed state over the activated and desensitized state. And we showed that data to show that compared to the huge effect from the closed state binding, the binding to the activated state is pretty minimal. However, the way we did the experiment, that the window of the time for the peptide to bind to the activated state before the channel desensitization is maybe too short. If the activated state on rate is like that, similar to the closed state. Since we assess the activated state binding by simultaneously apply the peptide with the acid, but it is preferentially bind to the closed state at pH 8.
Thank you. Next question. What's unique about WRPRF that makes the channel desensitization kinetics so much slower than RPRFA?
It's a good question. So there are some possibility. So with the addition of the tryptophan, that may that it may increase the affinity for the activate state or the open state and stabilize it there, or it may provide an kind of a steric block that prevents the structural rearrangements necessary for desensitization. And also, alternatively, during activation, the binding site may change conformation. So it may kind of shut the and trap the tryptophan there and keep the WRPR bound very tightly while the WRPRF bound tightly while the RPRF can dissociate before it desensitizes. So I think a structure will be helpful, and also a structure will be helpful with the peptide bound, and also using it in combination with molecules that known to affect desensitization will be also informative in that aspect.
Thank you, Dr. Yang. Next question, do you provide qualification data to compare the ready-to-assay cell lines with their corresponding stable cell line?
The answer is yes. So we provide data sheets and C of A certificates of analysis for all of our stable cell lines as well as the ready-to-assay cells. And I think I might have had a mic problem on my last question, which was, are the ready-to-assay cells division arrested? The answer is yes, they are, using a proprietary method that we have. Thank you.
Thank you, Dr. Costantin. Next question is, are mice ASIC similar to rat ASICs in distribution and properties?
Actually, we have tested a couple mice, and I think it's very similar to rat as we see. It's only probably one out of 5 or like 10% or less. Then the neurons have ASIC, so that's kind of similar. But I would say that the distribution is a little bit hard to say with the amount of data we have, but I would expect it's probably very similar between rat and human. That's similar, but it's very different from between rat and a mouse, but it's very different from human.
Thank you. The next question, have you thought about doing mass spectrometry on the human DRG neurons? Top-down proteomics could confirm the stoichiometry, and bottom-up could provide insight on why there are phenotypic differences in the EFIS profiles.
No, we have not.
Thank you. And here is another question. Would it be possible to turn WRPRF from an activator into an inhibitor as a potential therapeutic?
That's a very interesting idea. So I think it might be possible that we're looking at the SAR, but we may need more information on the mechanism of the peptide of its effect. So I think peptides, you think about, it's generally very flexible, but if we can actually know or see how specifically how the peptide fits into its binding pocket, we could make rigid small molecule that could kind of arrest or lock the channel in a specific state that will turn into an inhibitor or activator. But I would say we'll need better understanding of the interaction from a structure. Ideally, that it's probably the best to guide this process, but I think that idea is theoretically possible.
Thank you very much, Dr. Yang. And with that, unfortunately, we've actually run out of time. So I just want to thank Dr. Yang and Dr. Costantin for a fantastic discussion. And thank you to everyone also joining us online and for submitting your questions. Any unanswered questions can be answered by email afterwards. As you leave the webinar, a short survey will appear on your screen asking you to rate your experience. If you're unable to complete it now, it will also be sent to you via email shortly, and we would greatly appreciate any of your feedback. The webinar will also be available to watch on demand. So on behalf of Drug Target Review and Eurofins DiscoverX, Thank you for attending, and we hope to see you again soon. Goodbye.